Hello, everyone. This is going to be a video on making punnet squares, but not only the actual making of the punnet squares, but where that comes from in terms of the DNA and the genome itself. So let's take a look at this chart or this table where we see some inherited genetic conditions in humans. And it just lists them categorized as either recessive or dominant phenotypes or genes. So we have albinism, cystic fibrosis, sickle cell. These are probably things you've heard of before, as well as Huntington disease, freckles, polydactyly, and dimples. So I want to focus on this one, polydactyly, and use this as an example. So this is the state of having 5 digits or more than 5 digits. So we use the term polydactyl or polydactyly to describe an individual that has more than 5 digits. And this can happen in humans, and this can happen in other mammals as well. So for example, my first cat, Rosie, had polydactyly, and she had more than 5 fingers and toes. You could see here, it ended up looking a little bit like a mitten, or she had a thumb. And it's not a harmful mutation at all, but this was in her genetics. And so let's take a look at the genetics and remind ourselves what exactly we're talking about here when we talk about polydactyly or any other traits. Sorry, just trying to rearrange my Zoom. Okay. So I want to emphasize that there are a couple of terms that we're using here. We are talking about genotype and phenotype. And so our genotype, is the DNA sequence. And then the phenotype is generated from the DNA sequence. So we go from DNA, we talked about this, we go to mRNA, then we go to protein, and then that protein has some function which will result in some phenotype or a visible or observable trait. And so there's a connection between genotype and phenotype, but sometimes, as we're going to see, there could be a little bit of a disconnection as well because of the different variations that we could have in a gene. So for genotype, as we've talked about, we can have dominant and recessive versions of the gene. And these are going to have 2 different phenotypes. But we also know that we have 2 copies of every gene, one from our mother and one from our father. So we can have a dominant version of the gene for polydactyly. And so I'm just going to make it an H for hand. So we might have the dominant version of the gene for polydactyly from mom, and then the recessive version from dad. And even though the recessive gene is there, it's not going to be shown. Whereas on the other hand, if we had two versions of the recessive, or two copies of the recessive genotype or DNA sequence, then we would end up seeing the recessive phenotype. And for polydactyly, the dominant version is actually the version that we might not think of because it's a little less common. So again, in polydactyly, the dominant, which we said was the capital H, that means that there's more than 5 fingers or toes. The recessive version is the lowercase h, and that means that there are 5 fingers or toes. But remember, again, no individual, no human being, no mammal is going to have a genotype that's just this. Because that would mean, think about it, in your cell, you would have chromosomes 1 through 22, and that's it. But we know that's not what you have. You have chromosomes 1 through 22 from mom, and then chromosomes 1 through 22 from dad. So we have all of these guys in pairs. And this one. Okay, and so that means we would have another version of the gene from the other parent. On the other hand, in reproductive cells, as we said, reproductive cells are going to split up the genome into 2, literally, physically. There's only half of the genome. So in a reproductive cell, you're going to take just one of these. So the resulting cells, instead of splitting the cell in half and ending up with the identical genome that the original cell had, in this case, for reproductive cells, you're going to have only half of the genome, which means that you get either the chromosome from mom or from dad, as well as the rest of the chromosomes as well, but only one from each parent. And we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. And you're going to end up getting a mix of chromosomes from mom and from dad in this one reproductive cell. And then the same thing will go for this one, but I'm not going to draw all the little dashes. Okay, so let's move on to actually doing that Punnett square. But it's really important to understand the background of this before you start doing the Punnett square, because we all make mistakes. And it's actually pretty easy to make a mistake setting up a Punnett square. And many students do, and setting up the Punnett square is the most important part. But if you look at your Punnett square that you've set up and you know the background here, then you're going to be able to see whether or not the Punnett square is set up correctly. So actually, I'm going to give each of these a color. And the dominant version is going to be yellow, and the recessive version is going to be green. So it's the same trait, that is polydactyly, the number of fingers, and we have 2 different versions, the dominant and the recessive. And our parents that we'll talk about, or our individuals that we're going to talk about crossing, one of them is homozygous dominant. And then one of them is going to be homozygous recessive. And I just made this up. And this should be given to you in any kind of problem that you go through. Okay, so if you wanted to set up a Punnett square to cross these individuals and predict the genes that their offspring are going to have, and therefore also the phenotypes, whether or not their offspring have a chance of being polydactyls and having more than 5 fingers or toes, you are going to diagram the reproductive cells that can be produced by each of these individuals. And in this case, a moment ago, I drew out all of the genes or all of the chromosomes in the genome. We don't have to do that, luckily. All we need to do is take care of this one gene that we're talking about. And so we know that the 2 options here are to have this or this. This parent can only donate the capital H that is the dominant version of the gene. And as we said, the one on the left might come from this individual's mother, or what we might want to call the grandmother. And then the one on the right comes from the grandfather. It doesn't really matter at this point. We just know that these are the 2 types of reproductive cells this individual can donate. And then similarly, this individual can only donate this type or this version of the gene sequence. And now we just have to line up these different reproductive cells in a Punnett square. Let's make a little bit of room for that here. And the individual on the left is going to have either this or this in the reproductive cells. And then the individual on the right, we'll put on the top. Doesn't matter, you could switch this, it would come out the same. And this is the setup for a Punnett square. And then as we saw in class, you can do this one of 2 ways. You could do the dominance first, or you could do the top first and then do the side. I think it makes sense to do do it in order. So you could do the side first and then the top or whatever. So I'm going to do the side first and apply this capital H, the dominant version, to both of these boxes. And then we're going to apply the lowercase h's. So this one is lowercase, this one is lowercase. And then we're applying this one, lowercase, lowercase. Great. So the offspring here end up being all heterozygous. Or all the possibilities are heterozygotes. And when you have heterozygous, you don't add the recessive or dominant. Heterozygous means one of each or 2 different things. And so each one of the offspring is the same as we see here. All of them have the dominant version of the gene as well as the recessive version of the gene, which means that that dominant version is going to be shown because it's stepped in front of the recessive version. The recessive version is stepped back, it is recessed. And in this case, for this particular gene, that means that all of these individuals are going to be polydactyls because the dominant version of the gene is to have more than 5 fingers or toes. Okay, so let's take this a step further and we'll say, okay, well, what if we have 2 individuals that are both heterozygous for this trait? How does that work out? And I want to introduce some terminology. So sometimes this is referred to as a trait skipping a generation, which means that we didn't see any individuals in that generation that we just saw, the first generation, we didn't see any individuals displaying the recessive phenotype. So none of the individuals had only 5 fingers and toes. All right, so now we have these individuals, which are both heterozygous. And now we want to see what type of reproductive cells they could produce. This one on the left, well, we could get the capital H from that sister chromatid, homologous chromosome, or we could get the lowercase h from that homologous chromosome. And then on the other side, we have the same thing going on. And then we put these into our Punnett square. Finish this. Okay. So again, one individual goes on the side. I'm going to put the left one on the side and the right one on the top. It doesn't matter which way you do it. Either way, it's going to come out the same. And in this case, both individuals have the same possibilities for their gametes, for their reproductive cells. And so again, we're going to go and look at the left side first and apply these across, and then we'll apply from the top to the bottom. So we have a capital H here from the left, and then we have a capital H here from the left, and then we have this, and that gives us a lowercase. Actually, it's going to give the lowercase there. It's easier if you put the lowercase second. So now we're going to look up at the top and bring that capital H down. And then that is going to go here as well. And then we're going to look at this this possible genotype, possible reproductive cell, and apply that here. So now we have the whole Punnett square filled out. We have 2 individuals who are heterozygous. And then we have 2 individuals who are homozygous, one of each. So we have the one on the bottom here is homozygous recessive. And then the one on the top here is homozygous dominant. And so the homozygous dominant has more than 5 fingers or toes. And then the heterozygotes also have more than 5 fingers or toes. So if we count those, we have 1, 2, 3. We have 3 individuals that are going to have more than 5 fingers or toes, or who are polydactyls. And then we have 1 individual who shows the recessive trait. So hopefully that clears up everything from here. And let me know if you have any questions.